Hi, welcome back to protein. It's pretty short also. Okay, so proteins itself, we have got, again, likewise, you have got carbon, hydrogen and oxygen, which is also in your carbohydrates and fats. Okay, but unlike these carbohydrates and fats, in proteins, all proteins contain nitrogen. Okay, now proteins essentially they'll be broken down into the smallest units, which are amino acids. So carbohydrates, the smallest units are your monosaccharides. Fats, the smallest units are your fatty acids and your glycerol. Proteins, smallest units are the amino acids. Okay, now some of these amino acids, we have got 20 in the body. So some of these amino acids contain sulfur and phosphorus as well. Not all of it, but some of them do. Okay, however, all these 20 amino acids do definitely contain carbon, hydrogen, oxygen, and nitrogen. Okay, now proteins, they are extremely important to the body system for growth and repair of cells over here. Also a source of energy. Okay, not much because most of the time we don't really break down proteins. It's the last source to be broken down. So two types of protein structures that the IP student will have to know: globular protein and structural protein. Now, as the name suggests, structural proteins means they give a structure to the body system. So things like the keratin in hair, okay, in muscles, they give us structure to the body. It consists of actin and myosin. Okay, the blood clotting consists of fibrin as well. Okay. Globular proteins will be your enzymes and antibodies that is going around in the blood plasma. So likewise to your carbohydrates and fats, we can also test for proteins using the Buret test itself. So Buret test, you have got a small amount of food sample. Sodium hydroxide is added and shaken, and thereafter you add your copper 2 sulfate. Okay, purple coloration indicates that you have got the presence of your proteins. Okay, but actually this purple coloration, why you have got this purple is because copper in this copper 2 sulfate solution that you added in step 3, the copper ion itself will form a complex, meaning to say it combines with the nitrogen atoms in the polypeptide chain. Okay, so essentially we are actually testing for peptide bonds that are present among all the protein component, but not actually for specifically protein itself. Okay, now amino acid structure, we will look at the table below at the bottoms to be more clear about it. We can see that the amino acid structure comprises of a few different parts. On the left side, I have got the amino group or amine group. Okay, on the right side, I have got a carboxyl group here. Okay, so remember we have got 20 different amino acids, but you are not required to know all the names of these 20. So often, this 20 we denote it by this thing called the R side chain. Okay, and remember that your carbon requires 4 bonds. So we have got 1 bond here, 1 bond here, 3 here. So the last bond here is bonded by a hydrogen, which is a standard one. Now, over here, I have got one amino acid denoted by R, one R group here, another R group here. Okay, so this part here, I have got my carboxyl group. This part here, I have got my amine group. So once the amine group and the carboxyl group undergoes a condensation reaction, okay, so essentially water is being lost, correct? Okay, so where water is lost, my H2O is being lost here. I will have this O and your hydrogen, nitrogen, and carbon combining together to give me a peptide bond here. Okay, so we use the Buret test and we test for the presence of the proteins. The copper ion essentially forms a complex with the peptide bond over here, which gives you an indicative that there are presence of peptide bonds which exist in proteins itself. Okay, now these polypeptides, as they get longer and longer in structure, they will form into a 3-dimensional structure which is convoluted. Okay, now this 3-dimensional structure which is convoluted amongst is in order to hold this structure together, in between these bonds, I will have things which are called hydrogen bonds. Okay, so these hydrogen bonds help to keep the shape together, but when I heat the protein up to be extremely high temperatures, I have got presence of acids or alkalis that changes the pH of the surroundings. These bonds are being lost. So once the shape of these bonds are being lost, the protein structure is no longer as it is, and therefore it is said to be denatured. Okay, so often the protein that we consume is in a three-dimensional form, but as you can see, it is too big to enter into the cell membrane. So what happens, they will have to be broken down into the smallest possible units, which are amino acids, and once it is broken down in the small intestines, it will be absorbed by the body to be utilized as it is. Okay, now for the last part, we're looking at essential nutrients and also water itself before concluding the chapter. Thank you for watching.